Hi, my name is Judith Klein-Seetharaman. I'm a professor at Arizona State University, jointly employed by the College of Health Solutions and the School of Molecular Sciences. I'll be talking to you today about the molecular modeling of Usherin disease mechanisms and interventions. In this slide, you see an introduction to the USH2A gene. Mutations in the USH2A gene are the most common cause of Usher syndrome. The structure-function studies of this gene are very difficult because of the large size of it. There's more than 5,000 amino acids, which is making the protein tenfold larger than a typical size of a protein in the human genome. Shown below is a schematic of the USH2A gene. You can see here on the left, the N-terminus, the beginning of the protein sequence. On the right is the C-terminus. And you can see that there's many different repeats of so-called domains. And the specific domains and their names are spelled out here in the legend. Most importantly, I would like to point out the one here on the right-hand side, the yellow one, which is indicating the presence of a transmembrane helix. So USH2A is predicted to be a membrane protein, which means that it's located on the surface of the cell with a membrane anchoring it in the membrane, and so keeping it in place. And on the right-hand side of that yellow transmembrane helix is a very short segment that is facing the intracellular side of the cell. That segment has a so-called PDZ binding motif. So while this is not a big domain by itself, it is a motif that is recognized by many other protein binding domains and thus many other proteins. And this aspect actually serves as a collector for other proteins inside the cells that can then bind to the USH2A protein. On the other hand, on the left-hand side of the yellow domain, everything that is shown there, this huge 5,000 amino acid long sequence is completely extracellular. And so you can see that there is some domains here, for example, the laminin domain and laminin type domains. There's multiple repeats, but they're all facing the extracellular part, enabling interactions with extracellular matrix proteins. Of interest is also the location of one of the most commonly found mutations in Usher syndrome, the 2299 deletion, which causes a deletion and a frame shift at position 767. So what that means is that after this position where I place the arrow, everything else is missing. So only this very first piece of this protein is actually present. And that's really important because that means that there is no anchoring in the membrane. There is no PDZ domain in the cytoplasmic tail that would be able to interact with other proteins. And so this is really, really important feature of this mutation because it now fundamentally changes the function of this protein. And one hypothesis that we are proposing based on this drastic deletion of the vast majority of the protein sequence is that there is, on the one hand, a loss of function because of all this missing part, but there may also be a gain of function because the behavior of the segment of 767 amino acids at the beginning here in isolation is likely going to be different when it is by itself as compared to when it is embedded in the full protein. The second piece of information I'd like to present you with is actually the expression levels of this gene. So what you can see here on the left-hand side is the expression of the USH2A gene in the mouse and on the right-hand side in human. And what you can see here is that the expression is very specific in the eyes, both in mouse and human, and is very rod and cone photoreceptor cell specific. You see that there are some other cell types that have a little bit of expression, like here and here, but the amount is much smaller, so the color blue is much less represented in these other cell types. And so what is the effect of this disease mutation now on the USH2A protein structure? So we have analyzed the protein structure by using a program called AlphaFold. AlphaFold is a new prediction method that allows us to take a sequence as an input and predict what that sequence of amino acid building blocks looks like in three-dimensional space. And so what you can see here on the left-hand side is the prediction of AlphaFold for USH2A in the full-length wild-type unmutated structure. And you can see that it's predicting a big hairball. This is very problematic because it does not represent what we know about the membrane anchoring. So this is one of the limitations of the current prediction models, that while it can take into account what these large sequences in the extracellular domain are looking like, it cannot really tell you exactly how it is arranged on the surface of the membrane, because these prediction models are actually not taking the membrane into account. However, we can take a look at what is the predicted effect of the mutation on the protein structure, irrespective of the membrane. And that's what's shown here in the middle. When we introduce this mutation, which results in this smaller fragment that's only 766 amino acids long, you can see that AlphaFold predicts this to be a very well-structured, well-defined, relatively small protein. And so this indicates directly that there may be a gain of function in this protein, in that it is no longer embedded in this large amount of competing contacts that are made in the full-length protein. And so what you can see here on the right-hand side is the potential functional consequence, because we believe that the proteinaceous parts are making contact with other important proteins of the extracellular matrix, for example, collagen. Collagen is the most abundant protein in human and is a major component of the extracellular matrix and allowing to connect tissues to one another and preserving their stability. And so the interactions are really critical in both the formation and maintenance and remodeling of tissues. And so these changes that are predicted by the mutation would very likely interfere with the protein-protein interactions, both in terms of the extracellular side. But now remember that also this green part here at the bottom that's labeled with receptor is now missing. This part is actually no longer bound to the membrane. And so one possibility might be that this new fragment may be binding to other receptors and taking on the function of a ligand. What that means is that a ligand can then interact with a new receptor, and that would cause a new signal transduction cascade, which may tell the cells to respond to the signal of this mutated Usherin proteins in a different way than if it was embedded in the wild-type situation where it has no opportunity to interact with other receptors because it is embedded in interactions with the own long Usherin protein itself. So these are all hypotheses that can come out of these computational predictions that would have very profound significance for the design of interventions for this disease. And so one hypothesis that we have been investigating is, are there other proteins that might be able to compensate for the loss of function of the Usherin? And so our hypothesis is that such a protein should have similar interactions with other proteins. It should have a transmembrane helix to anchor it in the membrane. It should have a PDZ binding motif because we believe that would be critical for attracting other proteins to bind to it in the cytoplasmic site, and should perform similar functions as compared to the Usherin. And so the idea is that in the case of a mutation, maybe that protein could compensate for Usherin. And so one implication for therapy would be to activate that protein. And so what we have done is use the so-called guild by association principle. So there's a software called Guildify, which allows you to statistically compare different protein-protein interaction networks with respect to the similarity of those networks compared to each other. And that's what we have done by utilizing proteins that are involved in the Usherin protein interaction network. And we looked at it in the context of several other types of networks. For example, those associated with Usher syndrome in general, those associated with retinitis pigmentosa, those associated with oxidative stress, and several other critical functions. And what we have found by using that analysis is that there's multiple protein candidates that are listed here with their gene symbols. These are all candidates for proteins that have functions that overlap with the USH2A. And several of these are actually expressed in the retina, although the level of expression can be different. And a subset of these also have a transmembrane segment. So this gives us several candidates that we could investigate with respect to their similarity. And we are currently in the process of building structural models similar to what I showed you for USH2A, so that we can investigate the structures of these proteins and predict what kind of interfaces they might form with other proteins in order to verify whether these are good candidates for compensatory proteins that could make up for the loss of function of Usherin. So what we are currently doing, because we are so interested in the structural consequences of these mutations, is building new computational models. So the Usherin protein, because of its large size and complex structure, really poses a big problem to the protein structure prediction methodology. So the structure that I showed you that AlphaFold method predicts is a large hairball, which does not take into account the membrane. So one very important goal of our current research is to develop new prediction models that are optimized for membrane proteins that would allow us to predict a protein like Usherin with higher fidelity. So the way we are viewing the structural organization of Usherin at the moment is depicted here by this fisherman's glass piece. This is how we envision probably the Usherin protein would interact with the cell surface. So if you imagine the glass ball is the cell and the glass is the membrane, so the large extracellular portion would be sitting outside of the membrane, embracing it and having a major structural impact onto the cell. And we believe that there is interactions between individual domains of multiple Usher proteins. So although Usherin itself is already such a large protein, we believe that it will make interactions with other proteins. And in particular, we predict that these are trimer interactions because of similarities of other protein structures that involve the domains that I showed you at the beginning of my presentation. And last but not least, there's the transmembrane anchor. In this analogy image with the glass ball, this is overrepresented in size. Remember that these parts that are on the glass ball comparatively would be much, much larger than the transmembrane domain. But it shows you the idea in that the transmembrane helix takes on the function of a handle. And so it is the handle that allows placing it and keeping it in the membrane that associates it tightly with that particular cell. And on the other hand is the little cytoplasmic domain, and that is relatively accurate compared to the size of the transmembrane helix, in that it really only harbors the PTZ domain, which allows many different people that can attach to the handle in the same way many different proteins can attach to the cytoplasmic handle of the protein. So this is the model of the protein that we are currently working on. And this will allow us in the future to create much better models for the Usherin protein and its interactions. With that, I would like to acknowledge my collaborators. I would like to thank Agnieszka and Tony Ray for introducing me to Usherin and the Usher syndrome. I would like to acknowledge Gil Speyer from the High Performance Supercomputing Center that allows us to carry out these very intensive AlphaFold predictions, considering that the protein is such a large protein. This requires high-performance supercomputing. I'm currently working with Micheline Harris, who is my capstone student at NYU Informatics Protein and also works at Microsoft, who is working on the structure prediction methodology. And Baldo Oliva, who is my collaborator at Universitat Pompeu Fabra, who is the developer of the guild-by association predictions that I showed you. And last but not least, my two experimental collaborators, Paco Munoz Lopez, also at the Universitat Pompeu Fabra in Spain, who will test the predictions that we are making in cell lines representative of Usher mutations, and Susanta Saka, a single molecule biophysicist, who will visualize the interactions that the Usherin protein can make with cells. Thank you very much.